Welcome to Blueprint of Life video 4. In this video, we're going to be again looking at some evidence of evolution, and we're going to be looking at 5 different areas, and then we're going to do a quick introduction into the investigation that you're going to be undertaking. So in terms of the evidence for the theory of evolution, there are 5 categories which we're going to cover. We're going to look at paleontology, or the study of fossils and transitional forms. We're going to be looking at biogeography, comparative anatomy, comparative embryology, and biochemical analysis. The first of which is paleontology, and this refers to the sequence of fossils found in rock formation, and we know that the order of changes will be observed in these rock formations, and we can find in those rock formations that organisms may have originated from a common ancestor. In the fossil record, we also know that the fossil record should show intermediate forms. So allow us to see the transition from one organism to the other as it went through the process of evolution and natural selection. The evidence which we have to support this idea are fossils in undisturbed rock show a similar sequence, and we use transitional forms as fossil evidence. I've listed 4 examples here. So there's the Chrysopterygii, which is a lobe-finned fish, which shows the transition from aquatic organisms to amphibians. So they have a lobed fin, which might have allowed them to drag themselves along riverbanks as the environment became drier. There's the Archaeopteryx, which is a change from reptiles to birds, and we have some organs which we'll look at in the next slide. Whales have a similar, and horses changes in their hoof structure can show changes in the organisms from a common ancestor. So here we have the picture of the Archaeopteryx. So we can see here that there used to be a feathered wing here, but it's got finger claws. We've got vestigial organs around, a bony tail, all those kind of things. Just as an example, some limitations of using paleontology and transitional forms is that the fossil record is sometimes incomplete, and that bias is lent towards organisms that are better suited to fossilizing and becoming fossilized. So therefore, there's an unequal representation of some transitional organisms compared to others. And we know that dating of fossils is sometimes really hard, as radiocarbon dating only allows us to date fossils up to 50,000 years old, which might seem like a long time, but is not a long time when we're dealing with the evolution of species. The second is biogeography, or the study of the geographical distribution of organisms that are both living and extinct. If isolation is criteria for new species, the new species should resemble species with which they share a habitat. And in terms of evidence for biogeography, the macroevolution of flightless birds and continental drift. So presently, there are a range of flightless birds. So there's the kiwi in New Zealand, the emu in Australia, and then a range of others. And their distribution around the world suggests that these birds originated from a common ancestor, which would have been on the Gondwana continent, which we learnt about in Life on Earth in the preliminary course, and that different populations have evolved on isolated southern continents as they drifted apart. So they once had a common ancestor, but as they've moved into isolated pockets, so Australia being isolated, New Zealand being isolated, they have in fact evolved to become different. Some limitations of this method is that it's limited to the species which have become isolated in time, and therefore those which we know were isolated, and is only useful for organisms that have undergone that type of natural selection. Thirdly, we have comparative anatomy, and this is the group that relates to the first-hand investigation which you're going to carry out. So it is the study of similarities and differences in the structure or the anatomy of living organisms, and this can be used to determine evolutionary relatedness. If organisms are more closely related, then they should be more similar in structure than organisms that were separated further back in time. There are two bits of evidence which we have when we look at comparative anatomy. So we look at homologous structures or structures that are similar, and they provide evidence of divergent evolution. And one example of that is the pentadactyl limb, with penta meaning 5 and dactyl meaning like the digit, so 5-digit limbs. So they once were one structure and they've diverged to perform different functions. And then there's analogous structures which have evolved due to convergent evolution, so they perform a similar function. And we see that in the wings of grasshoppers. And we also see that in eyes of fish and octopus and other different reptiles, depending on the environments which we're living. Here we have the pentadactyl limbs, which we were talking about. And some limitations with this is that some fossils are incomplete, which means that they lack this specific limb, which makes it different to compare the anatomy of structures in living organisms with extinct forms. And there are some superficial structures which show similarities, but may be analogous, and then they can cause some confusion when people are looking at them to analyze them and see whether they are in fact similar or different. The fourth one we have is comparative embryology, and that's the comparison of the developmental stages of different species. And species that are related show similarities in their embryonic development. In terms of evidence, we have a picture here of embryos which show similarities in their early development. The blue here is indicating gill slits, which is one thing that we look for in embryos, and the other is a tail with distinct muscle blocks that appear during early embryonic life, and these indicate a common ancestor. And the fifth one is biochemical evidence, and this biochemical or this biological evidence evolution involves looking at DNA or amino acids in proteins, sequence base pairs, and compare them to see whether organisms may share an evolutionary relationship. Differences in sequences can indicate changes that arose during the process of evolution, and sometimes this allows us to, in fact, change what we know and change relationships which we once thought we knew about to because they're different, because we now have this biochemical evidence. In terms of technology used, we're going to be looking at in video 5, but just quickly, we have amino acid sequencing, DNA to DNA hybridization, and DNA sequencing. When we're looking and using this method as a form of looking at evolution, species that have fewer differences in their DNA or amino acid sequence are in fact more closely related. An advantage of using these technologies is that we can compare organisms where homologous structures are not available. We're provided with quantitative results, so computers do most of the work and provide biologists with a number, which allows it to take out human error and is giving us, in fact, real numbers that we can work with. And we can reveal more detail in DNA sequencing. But in fact, it still has limitations because some DNA structures or amino acid sequences can revert back to their original form, so then the changes go undetected, and the techniques are quite expensive and costly for people to undertake. That's it for video 4. Make sure you tune in for video 5.